we do want to actually be able to explain what Mendel was able to physically see. He was able to physically see different forms of what he called characters, we call characteristics or traits, and that when we crossed forms, sometimes one form was hidden. So the recessive form is sometimes known as the hidden form, because when I cross the dominant times the recessive, the F1 is always the dominant version. And so here, he always saw purple. When he crossed these purple times themselves, that's when he would start to see 705 purple compared to 224 white. So Mendel gets this data, and he's looking at this data going, purple times white isn't light purple. It's not half purple, half white. It's always purple. But then when I make these two purples, then all of a sudden a small amount turned back up white. And so he continuously got this ratio that was very similar to 3 to 1. Keep in mind, F2 is F1 times itself. So a cross of F1 and F1. So we actually are able to understand genetics a little better because there's a lot that we know that Mendel didn't know. So when he got this data, he was only able to come up with some limited explanations for what he was seeing. What we know is that DNA is inside of cells. And DNA is divided into sections called genes. And that those genes code for amino acids in a certain order, and that's a protein. And that protein actually gives us a physical appearance. Remember all the jobs of proteins? They can be enzymes, they can be pigments, they can be structural parts. All of those different jobs of proteins. So we know that. And we actually know that our DNA is arranged into chromosomes. So coiled chromosomes is what we inherit. How do we inherit it? Via meiosis or meiosis. And so we actually understand this. We understand how parents are passing on genes to their offspring. And we do understand that those genes come in different forms. That depending on the DNA sequence, we can have different forms of a gene. So this really makes a lot of sense to us because we can look at it on a cellular level. The other thing that's really helpful for us is something called the chi-square goodness of fit test. And this is a statistical test to see if the observed data is similar to the expected. Do I see what I expect to see? I've made a hypothesis. I think that this is dominant and this is recessive. Is that actually what I'm seeing? And I'm not going to get perfect numbers. I'm not going to get 300 to 100 because things in the environment happen. So maybe one plant died, and that's why I have less. Maybe a raccoon ate one plant. So we do have to be very aware that we now have ways to statistically analyze if what I expect to see and what I observe are similar, statistically math-based similar. So even without knowing all of this, Mendel actually still derived 3 important principles, and this is kind of the big takeaway for today. So we're going to try to kind of get through these 3 principles. The first principle is the principle of dominance. And the word dominant actually works very well here. Dominant I think of as stronger. So his principle of dominance is that idea that green times yellow will actually give me all green. So what this means is that one version or one allele, here's this important word here, one allele is able to hide another version. So a great example of this in pea plants is short versus tall. If I have two tall versions, of course I'm going to see tall. If I have two short versions, of course I'm going to see short. But if I have a tall and a short, all that I'm going to see when I look at the plant is the tall. The fact that the plant grew to short and kept growing to be tall. So the principle of dominance is this idea of being hidden but not disappearing. And the fact that this hidden can still be passed on. So when it's being hidden, we actually call that organism that has a hidden a carrier. And so we have actually started to assign the versions letters. And so when we look at Mendel's principles, we talk about dominant and recessive, we go ahead and give dominant versions a capital letter. And recessive versions a lowercase letter. What's up with these letters? Why are we using these letters? Well, remember, when we talk about alleles, we're talking about DNA sequences. Well, I don't want to write out A, T, 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 C, C, G, whatever. You could make up whatever letters you want here. I don't want to write all of those different. And if I have A, T, 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 C, C, G, I don't want to write all of that out every time. So I'm going to abbreviate those versions with a letter. The stronger version gets a capital letter. The weaker version, the version that can be hidden if a dominant is there, gets a lowercase letter. And so what we have to start looking at is, what does the actual physical appearance look like? And so we're really going to have to kind of get into this idea of, what are these letters? Why are there two? What all that Mendel knew is that something can be hidden and it can turn back up. So there has to be a stronger version and a weaker version, and it's being hidden. He also knew that the parents were both involved. Each parent is donating half, and this is called the principle of segregation. We can think of this as separation, right? We know the word segregate. We can think of it as separation. And this, we know, happens during meiosis, when we separate the homologous pairs. And we reduce to go to haploid. Now, Mendel didn't know anything about meiosis, but he did know that each parent was donating half. He knew this because the hidden form can reappear. So it didn't disappear, it didn't stop to exist, it wasn't destroyed by the stronger form. It was still there, and it could be passed on. And so this principle of segregation we understand as those homologous chromosomes. That you got one from your mom, and you got one from your dad. But all that Mendel knew is that when you got your chromosomes, you were then able to pass on. So in the next reproduction, you were passing on something that sometimes didn't show up in your own physical appearance. So those two purple flowers that had some white offspring, neither of them are white. Where did that white come from? Oh, it was being hidden. And it had to be coming back from these parents. So the purple one didn't win. The white one was just being hidden in the offspring, and the white came back. The third principle that Mendel had is the principle of independent assortment. the fact that traits are inherited independently. So this diagram up here at the top of the notes is super important because it actually shows all three of Mendel's traits. Green times yellow, green, that's dominant. The idea of each parent donating half, looking at what we call the genotype, seeing that each parent actually did give part of its genes. And then this last part that Mendel noticed is just because a pea plant had green pods didn't mean it would always have green seeds, that those traits were inherited independently. So the principle of independent assortment is that different traits are inherited independently. And so we understand this because different genes that code for traits are on different chromosomes. So that's why it makes sense from a molecular base. That the fact that not all of your genes are on one big giant chromosome, that they're on different chromosomes, and that they actually cross over and remix with each other. So the basic idea is that one trait doesn't determine the second trait. Now, of course, what we're going to see as we go through is we're going to see exceptions to all of these rules. And that'll be in what we call non-Mendelian inheritance. This is where we're going to stop today and really try to make sure that we know these three principles.